The development of personalized medicine is one of the core objectives of the different programs done at the Príncipe Felipe Centro for Investigation. The sum of procedures which make up part of the creation of new pharmaceuticals are completely developed in the center, famous for being one of the 50 specialized centers in its field in Europe and one of the 4 existing nodes in Spain.
We are approaching the drug discovery process almost considering the whole scenario, from identification of targets, validation of that targets in plates, in vitro tests, with tests that are capable to be processed or to be carried out in a high densities and high throughput screening approaches. We have several libraries that we are able to run throughout these tests to identify some hits. These hits we are also capable to transform in lead compounds because we have an organic chemistry lab, not only at the small molecule processes but also at advanced therapies, for example, developing some nanomedicines from these lead compounds.
The support from the platform is equally applied to in vitro trials, working with research animals, and later in clinical trials. This allows the center to validate which therapies are the most adequate for a particular patient.
The drug discovery process is a very complex process which involves different phases, and these different phases require different technologies. We can cover from very initial stages in which we try to uncover the mechanisms of pathology. So once we identify these targets, we try to identify molecules which interact with these targets to see if they have therapeutic potential. We can combine all these technologies with more in vivo or in vitro technologies, even combine these with animal models to see whether the products we are identifying in the initial stages have this therapeutic potential in culture and in animal models.
All the protocols that experimental protocols in the animal care using experimental animals, they have to be approved by the ethical committee for animal welfare.
One procedure that is done in the animal laboratory has to do with the study of how tumors develop.
We take samples from a human patient, we expand those samples in animal models, and then we combine this with the use of many different technologies, from genomics to proteomics, nuclear magnetic resonance, several other techniques to characterize the tumor and to try different therapeutic approaches.
You can follow up for several weeks or months the development of those tumors, or how that kind of target that you just, or drug that you can block this cancer could just reduce the size of the tumor.
The effectiveness, which validates the beginning of the following procedure, the clinical trial with patients.
From patient samples, we are able to extract or to work with stem cells, with cells derived from patients, and to validate which therapies or which treatments are able to be the adequate ones for the patients, looking at personalized medicines.
Degenerative diseases, the development of tumors, or the study of rare diseases are some of the pathologies this platform focuses research on.